associate professor in the veterinary college in Slacey. So today we planning to discuss about Retroviridae family. I'm sharing my slide. I guess all of you can see my slide, and I'm giving in the full screen mode. Can anybody tell me, can you see my slide and can read it properly?
Yes, sir.
Okay. Okay. As you know, the network everywhere is not equally good. So I'm just closing my video link. On time, I'll on it again, and I'll continue in the audio mode if you don't mind. So today we'll be talking about and discussing about some aspect of the Retroviridae family. It's an quite interesting and a unique group of viruses for me and everybody to recognize, because they had certain very unique characteristics, and the name itself, the retro, derived from reverse transcriptase. Okay, initials of reverse RE and TR from transcriptase has been taken and given the name of Retroviridae, because this group of viruses, they possess the reverse transcriptase enzyme. These enzymes is nowhere available in any life system except this retrovirus and HepaDNA virus. So this is a very unique characteristics in the evolutionary process this group of viruses has developed. And using this enzyme, the main purpose of this enzyme, reverse transcriptase, is to go in the reverse directions, in backward direction. Like whatever we understand that from double-stranded DNA, utilizing DNA-dependent RNA polymerase, the RNA messenger RNA molecules are transcribed, but the reverse transcriptase enzyme did in the reverse way. Taking the RNA as a template, they can synthesize double-stranded DNA in the reverse direction. That's why they are called as reverse transcriptase. And the name of this enzyme is RNA-dependent DNA polymerase. So this is a very unique enzyme possessed by this group of viruses, and because of which they have certain very special characteristics that we are going to learn in the subsequent slides. And then we'll talk about some of the important diseases as per your syllabus recommendation. We'll talk about some of the diseases. Of course, there are many diseases on the Retroviridae family. It is not possible to discuss all within this session. So we'll give you guidelines to understand you about the Retroviridae family. Okay. So some of the words like we have this lentiviruses, means slow. This whole family included two important subfamily, Orthoretroviridae and Spuma. Under Orthoretroviridae, the different genus is alpha, beta, gamma, delta, epsilon, and lenti. The highlighted one, the blue highlighted one, is as per our syllabus. We are going to discuss it. So this member contains a lot many infectious agents, which produces very peculiar kind of disease, including tumors or tumorigenic infection. As you know, many members of retroviruses can produce tumor in animals and human. We have heard about the lymphosarcoma in human, HTLV infection. So that's a retrovirus infection. Now it is established that the retroviruses, along with some other viruses, can also produce malignant tumor in animals and human. So these are some of the unique things that we are going to discuss and learn in this particular session. So we'll be concentrating on alpha retrovirus, the avian leukosis, we'll discuss it. The Rouse sarcoma, as you know, this is the first tumorigenic infection identified to us. Then we'll restrict to the bovine leukemia virus, which is a very close associate of human T-cell lymphoma disease, HTLV infection. So there is speculation that this bovine leukemia can also infect and cause recombinations with human T-cell lymphoma disease. That's why it is important for us and is detected in our country also. And under the lentivirus, we are going to discuss about equine infectious anemia and Visnamedi infection. So in total, these 4 diseases we are going to discuss. And I guess all of you know the lentivirus include the HIV 1 and 2 infection in humans. So these are the things that we are going to learn in this particular session. So let me tell you, see, we the teachers in the university, we simply act as a facilitator. We help you to understand the subject, but reading and understanding the subject is your part. So at all time, you are free to contact me if any queries, any issues you face it during studying the textbooks and collecting the information. So don't hesitate, please contact. We are always available for you to clarify your doubts and so that you can understand the subject. Okay. Another my humble request is that although I am putting the slides and I'll be sharing the slides with you, but don't restrict to the slide itself. Please read the textbook and try to understand the concept very clearly and be thorough so that you will be confident in your life. So coming to the unique properties of retroviruses, why I say all the time, see, this is very, very unique viruses, because they are the example of diploid genome, which is the characteristics of the eukaryotic genome. They're having the 2 copies of each gene, right? Whereas all other viruses are having haploid genome. This is a unique characteristics of retroviruses. And later on, it is found that this retrovirus, in the evolutionary process, it is hypothesized that they probably derive from the eukaryotic genome only, as because many retrovirus genome is having very close similarity with the eukaryotic genome. So as such, in the later part also, I will understand that how they are having certain common genes. So they are enveloped and very 3-layered particles. The icosahedral symmetry is inside, and then they are having additional layer, protein layer, and then the envelope. So 3-layer covering are there. The inner core is a positive sense single-stranded RNA, very small genome, around 7 to 12 kb is the size range among the members. So it's a very small genome size. And it is a positive sense single-stranded RNA. I guess all of you understand about the positive sense. Let me remind you once again. Positive sense RNA are those RNA where the virus transcriptions, initial transcription doesn't occur. The viral RNA itself acts as messenger RNA and protein synthesis can initiate. Whereas in case of the negative sense RNA viruses, the transcription is must. So that is why they carry the special enzyme, RNA-dependent RNA polymerase. Okay. Although it is a positive sense RNA virus, but their replication strategy is quite different. They do not follow like the other positive sense RNA viruses that we'll be discussing in the second slide. So another very, very unique things, as I said, they possess the reverse transcriptase enzyme inside the virion, inside the core area of the virion, where the nucleic acid is attached with almost 30 molecules of reverse transcriptase enzyme. When a virus replication completes its cycle within a host cells, at the phase of assembly, they pick up around 30 molecules of reverse transcriptase enzyme and they come out of the host cell. Why they do it? Because they need this enzyme when they infect another cell. So this enzyme is not available in the eukaryotic cell. That is why the virus have to carry this enzyme in their nucleocapsid. And as such, the viral genome codes for this particular enzyme. It is not available in the eukaryotic genome. So this virus, this is another enlarged picture of the entire structures of the virus. So although this is in positive sense, but doesn't act as a messenger RNA, here replication strategy is quite different that we'll discuss again. Let us come to some of the unique characteristics of this RT enzyme, reverse transcriptase enzyme. So this is a very, very peculiar things that we see among the viruses. One protein with multiple function. Generally in eukaryotic system, it doesn't happen. One protein, one functions, no. But here, this RT enzyme, it acts as a RNA-dependent DNA polymerase. It acts as a DNA-dependent DNA polymerase. It also acts as an integrase so that it can integrate with the host cell chromosome as provirus stage. As well as it is also having an RNase activity. It can break the small RNA molecules. So multiple functions lies in the different part of the RT molecule. So this is a unique thing you must remember. The RT enzyme is having multiple function, and viruses with one enzyme they carry out the whole process. So let us take a review about the whole replication process of reverse transcript, these retroviruses. So this retroviruses, okay. These retroviruses, they once they attach to the cell surface through certain specific receptor. For example, the HIV virus, they prefer the CD4 surface molecule in the T helper cells. And likewise, varieties of cell surface receptors are selected by different categories of retroviruses, and initially they bind with the cell surface. This is called as attachment. Then, once this attachment will occur, that will trigger the penetration process. Lot of cytoplasmic changes will occur, and gradually the virus particle will brought inside the cytoplasm within an endosomes. And then within the endosomes, the capsid layers degrades. Okay, the endosomal enzymes can break this capsid layer. The viral nucleic acids will be released. It is double-stranded 2 molecules of positive sense RNA. But instead of transcribing and translations, this RNA molecule utilizing the reverse transcriptase enzyme, they will convert it to a double-stranded DNA with long terminal repeats, LTA regions. Both at the 5' and the 3' end, you will see there are certain terminal repeats, repeated sequence are there, which are very, very essential for establishment and attachment to the host cell chromosome, as well as for gene expression. So once this double-stranded DNA with the long terminal repeats will form, then gradually under the influence of the RT enzyme, it acts as an integrase. It will integrate with the host cell chromosome. This is the host cell chromosome schematic diagram is showing. Here they will integrate it. This is a very, very unique things you will see in the life system. That's why we say the viruses, they are not living, they are at the age of life. So we call them as the virus particle, right? So no other organisms can have this kind of properties. That's why it is difficult to term the virus as an organism. So rather we say the virus are certain particles, but they have the properties of an organism because they can produce the progeny viruses. So when they integrate with the host cell chromosome like this way, this stage is called as provirus. Please note it down, provirus status. And in fact, there are certain retroviruses, they may remain in the provirus stages ever long. Even the animals will die in this life without production of disease. The provirus status will be there. But under certain influences, this provirus, from the provirus, the transcriptions and translations that occur under the influence of the cellular enzymes as well as the viral enzyme, and new virus particles are formed. So when the new virus particles are formed within the cytosols of the infected host cells, they will acquire 30 molecules of RT enzymes inside, and they will come out so that they can utilize this enzyme when next time they infect a cell. This is the process how the retroviruses, they complete its replication. So integration of the retroviral genome to the cellular genome brings a lot of problems in terms of it can induce a lot of mutation. As a result, it can cause tumor formation or uncontrolled proliferation of cells. So that will come to this slides. Like many retroviruses, they can induce tumor formations or they can produce cancers. So all the methods of cancer formations can be collectively discussed in these 3 categories. There are 3 types of retroviruses. One is called as transducing retroviruses. This group of retroviruses, they possess a oncogene called as VONC. They possess an oncogene. We call it as a VONC gene. So this V-oncogenes can trigger the uncontrolled proliferation of cells once they get integrated with the host cell chromosome. And this type of retroviruses, they are termed as rapidly transforming retroviruses. They can produce the tumor formation very, very rapidly. Whereas the other two types of retroviruses, one is called as the cis-activating retroviruses and trans-activating retroviruses, they does not possess the viral oncogene. Rather, integration of the retroviral genome to the host cell as provirus can trigger the uncontrolled proliferation of cells and tumor formation takes place. Okay, so we'll go a little bit deeper insight to this process. So what we need to understand, the fully competent retroviruses, they have these 4 important genes: GAG, POL, PRO, and ENV. ENV is the envelope glycoproteins they coded. POL is the RT enzymes coding genes. PRO is the viral protease enzyme, and GAG is responsible for group-specific antigens, mainly the capsids proteins they codes. Okay, so this genes in proper orientation, when it is present, we call them as the normal retroviruses or non-defective retroviruses. However, the retroviruses which carry a viral oncogene, VONC, this VONC gene, the VONC gene usually remain within the ENV gene of the normal retrovirus. As a result, this retrovirus become defective. There is defective ENV genes, since the VONC gene is located within the ENV gene. Okay. So this type of tumorogenic retroviruses, rapidly tumorogenic retroviruses, are termed as defective retroviruses. Defective in a sense, they cannot produce their envelope glycoprotein properly and cannot comes out of the host cell as a virion, infectious virus particle. They always need the help of a replication competent and normal retroviruses. That means 2 retrovirus presence is essential. One is defective, which is having the VONC gene, and other is the normal. I mean, the missing gene in the ENV is compensated by the normal replication competent retroviruses. So we are going to discuss about the ALV. Of course, ALV doesn't have a VONC genes. Simply they can stimulate the cellular oncogenes. We'll come to that point again. So here I try to show you 2 aspects. This is a normal retroviruses, means replication competent retrovirus, which is having the GAG, POL, and in proper order. They can synthesize their own proteins and new virus particle can form. However, you can see this is an oncogenic retroviruses or rapidly transforming retroviruses where the VONC genes they carry, and this is located within this region. As a result, this is defective. So a defective virus always need a helper virus to complete this replication cycle. This is very, very unique things that we can see among this group of viruses. However, there is one exception. This is the Rous sarcoma virus, which is a tumorogenic retrovirus infections. They contain a gene called as the VSRC, but location is outside this GAG, POL, and N. As a result, the Rous sarcoma virus is an exception. This is a replication competent tumorogenic retrovirus, rapidly can produce tumor. Okay, and interestingly, you will find this VONC gene they acquire from the cellular gene of us. All the animals and humans, we are having some oncogene termed as the C-onc gene. That is called as the proto-oncogene. We are having certain oncogenes. We call them as the proto-oncogene or C-onc gene. This C-onc gene also the name has been given, although the name has been given as the oncogenes, but they are not producing any tumor formation. Rather, They control the cell cycle growth. They assure that the cells normally grow and divide, and all the regulatory things of a cell proliferations is controlled by proto-oncogene. When this proto-oncogene is mutated by insertion of provirus nearby this proto-oncogenes, this degradation occur and the cell undergo uncontrolled proliferation, what we call as the tumor formation. Let us try to understand these things in a pictorial diagram here. Suppose this is a retrovirus, it's attaching to the host cells, and then the RNA is converted into DNA copy with LTR regions, and they inserted to the host cell chromosome. It has been seen that the retroviruses, the cis-activating transforming retroviruses, their integration is very near to the cellular oncogenes. Again, I'm repeating, the cellular oncogenes are responsible for controlling the cell cycle growth in the normal circumstances. And this insertional mutagenesis leads to uncontrolled proliferation. It doesn't have B oncogene, but their insertion is just ahead of C oncogenes, the previous slide what you see. The third category is called trans-activating. This trans-activating is not specific to any location. Anywhere in the genome they can integrate, and they may or may not produce tumor formation. Under certain influences, sometimes they produces the tumor formation. So these are some examples of acutely and slowly transforming retroviruses. That's about the oncogenesis, like how these GAG pole and enzymes and oncogene insertion will cause to the insertional mutagenesis and defective acutely transforming retroviruses processes. And this is some sort of genetics understanding about the tumor formation. This is essential for us. Okay. So, dear student, don't get it scared. Like these things are very, very simple things. Probably first time if you read it, you may got some confusions, but after reading several occasions and discussing with us, definitely your concept will be clear. Okay. So again, I'm repeating and referring to this particular picture. See, like many a times we comes up with a lot of excuse. We don't have the net system. We don't have this. We don't have that. But I feel that all of you are surrounded with the excellent gadgets surrounding you. You have all system, and over and above, you are having everything is available and it is there in your possession. What is needed is that you must need an urge to know the things. This particular boy in front of this fast food restaurant where his mother works, he's just putting a table in the street light. He's trying to learn and read it. This is called the burning desire. All of us, we must have this kind of desire to know the science, since you are all enrolled to the veterinary profession. So you should be committed to yourself, leaving aside all these excuses. Let me tell you, most of the development and the discoveries takes place by the people without any machines, without any high-tech facilities. So everything is possible, only it is the mindset we have to make it up. Okay, coming to the diseases. First is the avian leukosis or lymphoid leukosis. This is the disease has been identified a long time back in 1904, I think 1904, and found to be some tumorogenic infection. So also called as a lymphoid leukosis. It is a neoplastic disease of chicken, and here the cellular transformation takes place. Okay, mostly the B lymphocyte transformation takes place. It is a visceral lymphomatosis condition. Because of the virus infection, the B cells having the surface IgM marker get transformed. Uncontrolled proliferation takes place because of this virus gene integrated in the process of cis-activating retroviruses. And the uncontrolled proliferation of the lymphocytes leading to formation of visible tumors and visceral lymphomatosis, were also called as the big liver disease. So this is an example of tumorogenic viral infection in poultry. So caused by this avian leukosis virus under the alpha retrovirus genus. So this virus, they may remain in two forms, exogenous and endogenous. This understanding is essential for you, exogenous and endogenous. So exogenous refer to the literal transmission. Suppose some birds are having infections and give the virus infection. So from another population, the virus is coming to this particular bird and can produce infections and can cause transformation of cells. We call it as an exogenous infections. However, one of the characteristics of retrovirus is that since they can integrate it with the host cell chromosome, they may also integrate it with the ovum and the sperm. If it is so, then the virus genome can be transmitted from the parental flock to the chicks. So in that case, the newborn chicks will have this copy of the viral genome almost everywhere in its body. So this type of infections are called as the endogenous infection. Okay, so endogenous infections because of the maternal antibody doesn't clear up for a long period, but they may transform at once they reach the sexual maturity and circulating or the protein antibody when goes down, then the tumorigenic infection produces. However, this type of exogenous infections which carrying this tumorigenic viruses, or they can activate by 6 activating mechanism can cause tumor. So this is exogenous and endogenous. Endogenous means vertically when it is transmitted and the copy of the genes remain within the cells is called endogenous, and exogenous is from lateral transmission horizontally. So the disease can be transmitted both horizontally and vertically, and we are more concerned about the vertical transmission because vertically transmitted, you don't get any clue that the bird is having any infection, and this will pass this copy again to the next generation. So somehow we have to control it for controlling the disease in the population. So different subgroups and viruses were identified. They are having antigenic dissimilarity, a lot of antigenic dissimilarity. As a result, we don't have effective vaccines to control this infection. Okay. However, good thing is that the virus is fragile in the environment, cannot survive for long. Once you inactivate the virus, then cannot survive and cannot cause diseases and cannot survive for long outside the body. So if you see the host, who are the host, then you'll find all chicken flocks. But in the commercial chicken, the mortality may goes up to 23%. But in normal other domestic chicken, it's sporadic cases only. We'll see 2 to 3% mortality is observed, not so severe. So once the tumor formation triggers, then only the mortalities appear in the bird. Okay, many birds they may simply harbor the genomes without causing any tumor formations. This is true with this infection. Okay, so transmission vertically and horizontally. This is the 2 ways. Coming to some of the information about how we can propagate the virus. So the virus can be grown in the chicken embryo fibroblast cell culture, and they do not produce any cytotoxic effect. Rather, they express certain surface glycoproteins like ZP51 protein. This can be detected by agar gel precipitation test once, or by immunoperoxidase test. We can detect the growth of the virus in the cells. Okay. There is another test available, but this is diagnostically not much important. Earlier, people tried an 11-dose-old embryo intravenously. The virus inoculum has been given. The hatched chicks will observe till it reaches the sexual maturity for formation with tumor. These are some experimental things only. Diagnostically, it is not important. However, we can grow the virus in the chicken embryo fibroblast cell culture. Then coming to the clinical symptoms and the pathology, then you'll find once the bird reaches the sexual maturity, then mainly we see the endogenous infections, or even the lateral transmission. If it occur, we could see at that time only. So it says that from parental flock, the immunity they purchase for a quite long period. And there are certain facts like sex hormones also play a role in controlling this disease. So certain experiments of by castrating the birds, caponizations, seen that the disease intensity is different. So clinically, if you want to see, then if the tumor formation starts inside the body, then gradually the bird become anemic, and that the weight loss will takes place. And then if death occurred in internal organs, you can see the tumors, visible tumors in the internal organ. However, in these infections, we never see any infections with the nerve tract, iris, and skin, which is a characteristics of Marek disease. If we can remember, in the DNA virus infection of the herpes viridae, the MARDI virus that causes the Marek's disease, which is also a tumorogenic viral infection in poultry, where one of the frequent outcome is the nerve infections, bilateral or unilateral these infections of the sciatic nerve, iris infection in the skin. These things are absent in avian leukosis, right? So basically, there is the tumor cells of the lymphoid organs, like the B cells marker, like 70 to 80% of the lymphocytes are the B cells affected, having the IgM marker, where the cellular transformation takes place. So the Bacillus fabricius is the main target, where in the bird the virus grow and tumor formation takes place. But later on, it disseminates to the body, to other organs like in the liver, gonads, everywhere they can localize and cause some solid tumor formation. Okay, so this is one picture is showing solid tumors in the internal organ. This is spleen, this is liver, and this is another diffuse kind of infections where we can see the tumor formation. All these white patches are the aggregations of transformed B cells, majority of transformed B cells. Coming to the diagnosis, how we can diagnose this disease. So obviously the clinical signs and symptoms gives us some idea about the postmortem lesions, but it is essential to confirm the disease, as because there are other tumorogenic infections like Marek disease also, this kind of things happen. Okay, so in live bird, even the cloacal swabs we can take it, and even the egg albumin we can screen it for presence of the virus antigen. More particularly while selecting the flock for entry into a farm premises. Okay, so there is one test called as the COFEL test. It is complement fixation test for avian leukosis. For quite a long period, people perform this particular test. But as you know, the complement fixation test is laborious and a lot of standardization is needed. Nowadays, we do not perform this complement fixation test for disease diagnosis. Alternatively, we are having other good tests like FAT, fluorescent antigen antibody test. So we can take any impression smear of these tumors and can stain with the specific antibody, and the infected cells can be detected by fluorescent activity. Okay. So earlier people also perform RIA, but as you know, the radioimmunoassay nowadays we are not using because of its hazard. But ELISA tests are extensively used for detection of antigens. We can go for isolation and detection simultaneously. This practice we can do it, right? Then the histopathological sections can see this the B lymphocytes, different types like immature, mature, and in comparison to the other blood cell population, the immature B lymphocyte populations will be very high. So these are certain aspects. And finally, like RT-PCR, it become very easier and very convenient for rapid detection and accurate detection of the disease by performing RT-PCR. So in the intensive market, certain very good antigen detection kits based on P27 protein is available, and this can be used even for screening the parental flock about the probable carrier status of the bird. So when you talk about the prevention and control, as the commercial good effective vaccines are not available because of many reasons, like the live virus vaccines we cannot produce because it's an RNA and integrated with the chromosome. Secondly, if you inactivate it, it will not be effective against endogenous infections. So these are some issues because of which the commercial vaccines are not available in the market. Some countries, they tried it as some recombinant vaccine. To some extent they are successful, but commercially it is not available in our market, right? So here basically we target to prevent the particle transmissions. The parental flock we should screen very properly. If any doubt is there, then we should remove the parental flock, and if we can break the chains of particle transmissions, then the disease can be averted. So this is the aspect. Now coming to the second infection, equine infectious anemia. Almost in 18th century, this disease was detected in France, and later on to other European continent and Middle East, and even in India in 1987, the disease incidence was recorded. So equine infectious anemia, it is a progressive disease, and in acute form, it may lead to very, very high mortality, up to 85% mortality may occur. So in such a high mortality in large animal is really a serious matter. However, the disease in other forms also, in the chronic form, the mortality is not so high. So another synonym of the disease is swamp fever, because this incidence of the disease is very much in the swampy area, low-lying, waterlogged area, and particularly in the rainy seasons, in the summer month, the disease incidence is more, and it is directly related with the vectors. So the disease is by arthropod vector, particularly the blood-sucking arthropods like tabenide and stomoxys flies. So they can literally transmit from one animal to the other very frequently. Of course, other mode of transmissions are also there. So mostly the virus remains cell-associated, and transmission of infected cells from one animal to the other, the disease transmission is very frequent, and it is very easily and conveniently can be transmitted by blood-sucking arthropods. Okay, so causative organism, it is a lentivirus. As you know, lentivirus are an example of slow virus infection, equine infectious anemia virus. So the host are equine species. Male donkeys are susceptible, although the serum antibodies has been detected in sheep, goat, and other species, but clinical infection has not been seen in other species. So if you go through how the disease spreads and among the animals, then you'll find majority of the cases it is by the mechanical transmissions by this blood-sucking insect, tabenide and stabulefly stomoxys. In swampy area, the number is more. That's why the disease incidence is more in those summer months in the swampy area. That's why the alternative name is the swamp fever. Another aspect is the iatrogenic spread. This is quite interesting. Like, like iatrogenic means the transmission through contaminated needles, syringes, and foam equipment, like AI gun or certain foam utensils. More particularly in the mass vaccination process, if you use a single syringe needle for vaccinating many animals, then through this contaminated needle and syringe, the disease transmission may occur. So this is called as the iatrogenic spread. Of course, this is very less. Then transplacental transmissions, in utero transmissions may occur, as well as direct contact, more particularly during the sexual contact, through the sexual contact. And the virus is mainly the cell associated, so cell transmissions is needed for transmission of the disease. So if you go through the pathogenesis of this infection, they are mainly the lymphotropic viruses. They infect the macrophages and the lymphocytes. And when the circulating antibodies are produced, the viruses, they escape the blood circulations. And again, the second set of viremia produces. Like this way, the animal, they exhibit some sort of ambulating fever. And most of the time, the immune complex get deposited on the surface of the RBCs. As a result, the complement activation takes place and the autoimmune destruction of erythrocyte takes place. So because of which the animal, there is progressive anemia productions, and the animals, they gradually die because of this anemic conditions. Okay. Similarly, the immune complex depositing in the glomerulus in the kidneys causes glomerulonephritis. This is again activation of complement. As you know, the immune complex always trigger the activation of complement pathways. So this activation of complements can cause glomerulonephritis, and that leads to death of the animal. So the different clinical symptoms, which is the characteristics of the autoimmune destruction of erythrocytes like anemia, jaundice, petechial hemorrhages, etc., can see splenomegaly, etc., etc. All these symptoms you can see in this particular disease. So coming to the diagnosis, of course, apparently looking at the animals, we can't get any idea. So confirmatory diagnosis is based on detection of antibodies as well as antigens. And for antibody detection, always we prefer the paired serum sample. You understand that paired serum sample means serum sample collected at 2 different intervals from the same animal. And if there is any increase in antibody titer, indicates an active infection. Because this is a slowly progressing disease, then the paired serum samples works in diagnosis of the animal. However, if you diagnose also there is no treatment available, only thing is to eliminate the animal from the population so that the other animals will be safe. So Coggin, the name of a scientist, he developed a simple AZID test for rapid detection of the disease, and it is famously known as the Coggin test. It's in 1970, I think, Dr. Lori Coggins. Yeah, this is an agar-gel precipitation test. I think all of you have performed it. Likewise, this kind of precipitations are produced in the central wells. We can give the antigens, and the serum we can give in the surrounding wells, and we can detect it within overnight time. And this is famously known as the Coggins test. Please note it down. This is relevant for diagnosis of equine infectious anemia. Besides this, the other tests like ELISA and FATs are also available for disease diagnosis. And the virus can be isolated in the equine leukocyte cultures and can be detected subsequently by the test, AZID test. So because of a lot of serological differences among the strains, the effective vaccines are not available to control the disease till now. Although I said that the recombinant vaccines were tried in some countries, but commercially it's not available. So it is the test and slaughter policy, we should adopt it so as to control the disease. So in India, after its first outbreak, substantially the numbers has been come down to a level of very low percentage by adopting this test and slaughter policy against this equine infectious anemia virus. Okay. Then the next is this ovine progressive pneumonia, VISNA and MEDI. Very shortly we'll discuss it. This has been detected in our country also. This is an example of slow virus infection. Please note it down, because it's, you see that the incubation period is 2 years. 2 years time is needed to develop the clinical manifestations. So this is an extremely slow virus infection, and because of which it has not been detected for quite a long period. And then the history in 1933 only, some ship from Germany was imported to Ireland, and then the ship, they developed this kind of gradual emaciations and wasting kind of symptoms they exhibited with respiratory difficulties. And they start giving the name as the VISNA and MEDI. This progressive pneumonia or MEDI, which means dyspnea. Basically, you'll find it that the growth of the virus in the lung tissues, particularly lung alveolar macrophages, causes thickening of alveolar septa. The alveolar septums are so thickened at the time of postmortem, when you cut open the thorax, the lung will not collapse. Up to that extent, the interalveolar septum thickness takes place, and that also after 2 years of infection. It's a very gradually occurring disease, and animal will undergo some sort of wasting condition, and that's what we call as the VISNA. These are 2 clinical forms of the disease. And this is because of nervine demyelination process. The virus also causes demyelination of cerebrospinal nervine tissues, and sort of incoordinations are exhibited by the animal. So the causative organisms, SIPs, are affected and can be transmitted by the direct contact with infected animals. Even the aerosols transmission also, it is speculated. And through milk, intrauterine transmissions, through milk and colostrum, the virus can be transmitted. Intrauterine is rarely occur. So it is again the cell associated. If the cell transmission occurs through certain injury, then the virus transmission may occur. However, droplet is an established mode of transmission of Visnamedi. So this is again the whole story of lymphocyte-associated viremia, and then central nervous systems are affected, as well as the lung. And these things already I discussed it. So do not collapse after opening the thorax. This is an important point to remember. So this gives us some information about this disease. So hind limb paralysis, perivascular coughing, these are some of the changes. Diagnosis, we can go for isolation since fascia formation takes place in the choroid plexus cell line, choroid plexus cell line. So easily we can detect it. However, it is difficult to guess the infected animals because the incubation period is very, very long. Gradually, the emaciations and shrinkage and dyspnea will produce, and finally they will die. So PCR-based tests can also be performed. Okay. So here also the same fact, the vaccines are not available for controlling the infection. Now coming to the last disease here, the bovine leukemia virus. I'm having 5 minutes time. So this is also called as the angiotic bovine leukosis. This particular virus is very closely related to the human T-cell lymphoma virus, and it is speculated that sort of recombination may produce among these two group of virus. They are under the Delta retrovirus. 7 genotypes were identified. So in India, there is some study conducted recently in 2018. Around 118 animals were screened, and out of that, almost 33 animals were found seropositive. And the samples, blood samples analysis was done by PCR method, and it found that the genotype 6 is prevalent in our country. However, recently again in the adjacent countries like in Myanmar, they recorded the genotype 7 from their cattle in the Myanmar. So this is an emerging type of disease, what I feel is affecting our cattle population. So this is transmitted by mosquitoes, direct cell transmission. This is quite important. So the cell-free virus rarely will get it in the animal. So animal rarely excrete the virus outside, or if at all, if excrete, the virus get inactivated readily. So the infected cells, if transmitted from infected animal to the normal animals, then the disease progression may take place. So possibility is the direct cell transmission through injections or blood transmission. Mosquitoes can play a role. Rectal palpitations, if any injury occur from one animal to the other animal, it may occur. Through broken skin also, if some cells transmissions takes place, then the virus may spread. Okay. The characteristics are similar to that of the virus is very fragile, cannot survive for long in the environment. This is the genetic structures. You can see this gag pole end is intact. There is no V on genes. So some of the genes like TAX, RAX, we'll not discuss about this gene. Their integration to the host cell chromosomes may activate as transactivating, and sporadically the disease outbreak may occur. Many animals may not show the disease in their lifetime. So once this transformation occurs, it is multicentric lymphosarcomas are produced, and the animals they develop certain posterior paralysis because of the nerve tract infections, and the animal they produces the superficial lymph nodes tumor formations can be noticed. This is quite visible, and the animal's conditions will deteriorate, and in the blood pictures and the postmortem you can see multiple this kind of tumor formation. This is in the cardiac, in the heart, and this is in the kidneys, this kind of tumor formation. And characteristics is the B cell transformation, the B lymphocyte transformation is seen. Okay, so SGPT is a recommended test for diagnosis. Besides, the peripheral blood monocytes from the infected animal can be cultured in the laboratory and can be detected by any one of these tests for confirmatory diagnosis. Similarly, the syncytia formation can be detected in fetal bovine lung cells. So this is the diagnostic approach, and over and above, we know like RT-PCR is always performed even for sequencing the particular genes like P24 or P51, GP51 gene. The phylogenetic analysis can be prepared to find out which genotype the virus belongs. Okay, let me tell you once again, in India, the genotype 6 has been detected in 2018 in such type of study. Okay, so no effective vaccines is available. Only the care we have to take it so that this kind of horizontal and vertical transmission doesn't take place. So it's mainly by insect and the lateral transmission of infected cells, lymphocytes. During the rectal palpitations, if some cells of the animals is attaching to your hand and without changing the gloves, if you go for rectal palpitation of another animal, the disease transmission may occur. So these are some informations that we gathered for today's session. So finally, what we discussed today is the unique properties of the retroviruses, the Retroviridae family, having the RT enzyme, how they can act as a pro-virus integration to the host cell chromosomes, and how they can induce tumor formation by integrating into host cell chromosomes. Some of them, they have the V genes, because of which they can rapidly transform the cells. However, the other do not have the V gene, but their insertion can cause transformation of the cells by cis-activating and trans-activating mechanisms. The lymphoid leukosis, as you know, like this is a tumorigenic infections, and how vertical transmission or endogenous infections are important that we discussed. About the equine infectious anemia, their transmissions and the severity, and Visnamedi, the example of slow virus infections, where it took a long time, almost 2 years time for development of the disease, and finally the bovine leukemia. But this is not the end of the story. There are many other diseases. Whenever you get time, please read book and try to understand about the disease. More particularly the HIV infection, please read it. Now you get the baseline that how the retrovirus behaves, and you understand the aspect of retrovirus replication. If any problems or anything arises, any doubt need to be clarified, please do contact me and other professors so that we can clarify your doubt and help you in understanding the science. Thank you very much. For the student has joined, I guess you have to join in some other classes. You can text me at any time, and the doubts can be cleared. Okay, thank you very much for today's session. Thank you.
Thank you, sir.
Okay.